Hi, and welcome to number 2 Edpuzzle study guide recording. So the first thing I'm going to do is I'm going to go over some commonly missed problems with the study guide, and then I'll go over some good test stuff with you. This will only take about 10 minutes, and that's it. So you see on the video, there's nothing there except a slideshow. So all you have to do is listen to this, stop it as you need to, and answer the questions. Let's go. So on the study guide, we see that we have the work of the following scientists. Make sure you know that they all use a compound light microscope. And then if we're talking about microscopes, let's go ahead and compare a light microscope to an electron microscope. One uses two lenses, known as the compound light. One uses electrons. Make sure you know the difference between the scanning electron microscope and the transmitting electron microscope. The transmitting electron microscope will actually go into the cell and talk about the surface or the dissection of the mitochondria. And a scanning electron will scan the outside surface of a cell so we can see the parts of the outside of the mitochondria or the outside of the cell membrane of cells. Then look at your scientist, Robert Hooke, did the cell term. Schleiden and Schwann gave us the cell theory. So what are the three parts of the cell theory? Make sure you know that. With the cell theory, go ahead and know that the cell is the basic structure and function of life. Many of us tend to forget that. Prokaryotic versus eukaryotic. Make sure you know that prokaryotic is bacteria. It's unicellular, does not have mitochondria. Therefore, that's why it does anaerobic respiration. The only example is prokaryotic is the bacteria. Different examples of eukaryotic cells would be plants and animals, fungus, anything that has membrane-bound structures. So then we go into the cell organelles and the cell parts. Make sure you remember the analogy of the cell to the cookie factory or to a factory, like the mitochondria is the powerhouse of the cell. But since we have learned cellular respiration, you know that if you see a picture of a mitochondria, it's going to be more into like oxygen comes into it and CO2 exits. It's cellular respiration that occurs there, specifically aerobic. So it's not just the powerhouse anymore, it's where cellular respiration occurs. So I'm reading through two organelles found in the plant but not in the animals, which reminds me. So the plant has the cell wall and a large vacuole and chloroplast. Animal cells has centrioles. You need to be able to identify what a centriole is. It looks like the little star part. So go to your book, look in the book to see the picture of the centriole so you can identify that. Plasma membrane we see, don't forget what those are composed of. The cell organelle that controls all activities is the nucleus, but make sure you know that it contains DNA, which leads us to letter E, DNA, heredity information. Assembles proteins, I'm on letter H, that helps, that's the endoplasmic reticulum along with the ribosomes. So that brings us to the two different types, rough and smooth. They're both going to be connected to the nucleus, and then the one that looks like it but doesn't have the bumps on it is the Golgi, and that's closest to the cell membrane because its job is exocytosis, which creates a membrane around the proteins and pushes it out of the cell membrane. Thus, the reason why it's close to the cell membrane. So then we go into in which organelle ribosomes produce is the nucleolus, so that was letter L, excuse me. Letter M, the organelle, it contains the green pigment chlorophyll. That's going to be chloroplast. Make sure you know all the structures of the chloroplast, just like we're on the quiz. Which organelle is responsible for changing glucose into usable form of energy? That usable form of energy is known as ATP, so this would be the mitochondria. Difference between cilia and flagella. Flagella is long and whip-like. Cilia is all over. Recycles old or unused parts, which is letter P. That is the lysosome that has enzymes in it. Then, how is cell like a factory? Number 6, we've done a 7. What is cell specialization? So, cell specialization, you need to think of stem cells. Stem cells are cells that don't have a job, so they are unspecialized. They are not differentiated. They are not expressing proteins to give them their job. So, cell specialization are cells that have a job and are expressing proteins that are important for that cell's job or function. 8 is what is a synapse? This goes back to the two ways that cells can communicate. One is by hormone, the other one is by chemical, which is going to be synapse. So don't forget this is going to be more along the lines of a neuron. Make sure if I give you a picture of a neuron, you know that it's a neuron and that its job is for communication. 9, the molecules found in the fluid mosaic model. Fluid mosaic model will be the cell membrane. So you have your 4 different types of proteins. You have proteins with carbohydrates for Communication meaning, I get confused with that, communication meaning that they identification with each other. Then you have the receptor proteins, which receives the hormone signal. Then we went into the plasma membrane and we looked at, okay, well now you have a gradient, a concentration gradient, which is where there's a different number of concentrations on either side of the membrane. That leads us to transport, active versus passive. Be able to look at these and determine what the picture is telling us. Is this diffusion? Is this water? Remember, if it's water molecules moving, it's going to be osmosis. All different types of passive transports. Active transport, the common two y'all get confused is a carrier, movement by carrier protein, which is active transport. You often get confused with that and facilitated diffusion. They have the same picture with the membrane and the protein, except you need to look and see which way the molecules are flowing. If it's moving from high to low using the protein, it is going to be passive transport, facilitated diffusion. If it's going against the gradient, it's movement by carrier protein, active transports. Number 15, it says what happens to a cell placed in a hyper, and isotonic solution. Like I said before, please don't worry about those terms. The only thing I want you to know is the beaker examples. If I give you a cell in the different types of scenarios, know which way water is going to flow. Therefore, an example, blood cell in water. Water is going to rush in, it's going to lyse the cell. Then you have on the top right, you have pictures of the animal and plant cells. Those are hard, I see. Number one is the cell, or the first blank is the cell wall. The second blank is the cell membrane. The third blank is mitochondria. The next one is only the animal cell. That is the centriole. The next blank right there is the chloroplast. Then that one is probably going to be the lysosome. Going to the next blank where it's talking about the folds on the outside of the nucleus, that is endoplasmic reticulum. Then the next blank, it looks like it's either the nucleus or the nuclear membrane. I will make it clear to you which one I would like. Then to the next one, it's pointing to the squiggly stuff. You might have nucleus there, however, I would say DNA. Then to the next blank, I should have numbered these, sorry. It's the little gray circles inside of the nucleus. Those would be the nucleolus. And then the next one, once again, that could either be nucleus or the DNA. Whichever one you didn't have two up is the one that you'd have there. The next one's pointing to the really big structure in the plant. That would be the vacuole. Then you have the next one, which is a little confusing in the fact that it's not close to the cell membrane on the animal cell, but it is on the plant cell, so that makes me think it's the Golgi. Then we go to the second to the last one, pointing to nothing except the background, which is the cytoplasm, and the little animal cell has the flagella, which is by itself. Then prokaryotes versus eukaryotes, lacks the internal membranes. That would be prokaryotic. Do not have a true nucleus, prokaryotic. Are either single-celled or made up of many. That would be eukaryotic. Most are single-celled prokaryotic. Organisms have cells that have organelles, are the eukaryotes. So due to time, I'm going to go ahead and talk about the test itself. So let me just try to help you here. Scientists have discovered a fossil. They also see that inside this fossil there's a double membrane structure that contains genetic material. Which cell organelle have they found? Probably the nucleus. If the molecules cannot pass through the membrane, what will they do? They will probably have some type of active transport by either bringing in a vesicle, like endocytosis, or they will use a protein of some sort. If I give you three different pictures and one has paramecium with these tiny little hair-like projections, those would be called cilia. If I give you an amoeba, which have all these little finger-like projections, that would be a picture of pseudopod. And then if I give you a picture of euglena and it has this whip-like tail, that would be flagella. Well, all those structures have to do with which type of characteristic in a living organism. It would actually be movement because they all aid in movement. What would complete the process of cellular respiration, meaning what are the products here? If I have glucose and I have, it goes through glycolysis, then what does it produce? Well, it's always going to produce CO2 and it's always going to produce ATP unless I tell you that it's anaerobic. So make sure you know how much ATP, so it'd be 38 here. Let's see, we're back now. Genes are made up of DNA. I know that's kind of review, and they're located in the nucleus. make sure you know that structure equals function, meaning if the cell is circular, it's probably going to go through tunnels and veins. If the cell is spread out and have all these dendrites, all these parts of the cell extending out from it, it's probably communication. Which cell has the most mitochondria? It would be muscle cells. Muscle cells are specialized for energy, while like sperm cells are specialized for movement because they have the long flagella. If I give you different examples of active transport, like if I'm comparing exocytosis to endocytosis, make sure you know that they are both using ATP. Endo would be out of the cell, exo would be out of the cell, endo would be in. If I give you a picture of a mitochondria, make sure you know that CO2 is leaving, oxygen is coming in, and that it would be aerobic respiration. If I tell you that the product is lactic acid and ATP, that would tell you that it's probably anaerobic respiration, specifically lactic acid fermentation. Duh. If I put a cell in a highly concentrated salt solution, means the outside has a lot of salt, what happens to my cell? Water would go out of the cell into the salt. Remember, I like to tell you salt sucks, but be careful with that because y'all tend to get confused with what salt does. Just know that where there's higher salt, that's where water is going to go to. Going back and looking at the hierarchy of cells, for instance, cells come together, the same group of cells, same structure, same function. They're going to make up a tissue. Tissues come together to make up organs. Organs make up an organ system. Which molecule is required for animal cells to obtain the most energy possible? From a molecule of glucose, it would be oxygen because that's the most efficient. So oxygen and glucose together will produce the most ATPs. The picture of the POD that we had, the bottle with the balloon over it, make sure you know that that is anaerobic respiration and that gas being produced is CO2. If we're looking at a Lodia cell, we see that the vacuole is real shrunk and is pulled away from the cell membrane. Please know that the turgor pressure has decreased and it will wilt, meaning it will bend down on itself. Be able to recognize all of them, all of the organelles, I'm sorry, I'm all over the place, all of the organelles. ATP, if I give you a picture of an ATP molecule, be able to identify that it is ATP and that the actual energy is stored in the last bond between the second and the third phosphate group. So if I ask you where it's stored and you circle the phosphate, it is wrong. It's the bond in between them. You're going to need to know the parts of the chloroplast, all the parts of the cells, be able to look at a cell and answer questions, not just say which is the nucleus. I might say which organelle is responsible in this picture for producing glucose. And you would say, okay, that's the chloroplast, and then you'd have to go to the cell and look to see that it's the chloroplast. All right, so good luck studying. If you need me, email me, and adios. Ciao.